Hi, I'm Ann Churchland, and I'm an assistant professor at Cold Spring Harbor Laboratory in New York. I'm going to tell you today about work I did as a postdoc in the laboratory of Mike Shadlin at the University of Washington. This work was part of a collaboration that brought together experimentalists and theorists who shared the goal of understanding perceptual decision-making. The title of our paper is Variance as a Signature of Neural Computations During Decision-Making, and the authors are myself, Ruspe Kiani, Rishi Chowdhury, Xiaojing Wang, Alex Pouget, and Mike Shadlin. We began by training the monkey to do a perceptual decision task where he made decisions about the direction of motion in a cloud of randomly moving dots. We then recorded action potentials from single isolated cortical neurons in parietal cortex, a part of parietal cortex called the lateral interparietal area, or for short, LIP. When we first began analyzing the data, we noticed, as many before us have, that the action potentials were highly variable from trial to trial. In this movie, I've shown 3 example trials, each shown by a different color, in response to a very similar trial, a trial where the motion was to the right and the monkey made a decision to the right. Each tick mark in the trials represents an action potential. And what is apparent is that the pattern of action potentials is quite variable from trial to trial, even though the monkey did roughly the same thing each time. To quantify this, we counted the spikes in small counting windows, which I've shown by these green boxes, and then computed the total spike count variance as a function of time. We then isolated the component of total spike count variance that is informative about neural mechanism. We call this quantity the variance of the conditional expectation, or for short, the VARCE. When we first began computing these quantities, we weren't quite sure what to expect. On the one hand, a previous study had computed a quantity very similar to ours, called the Fano factor, and had found that it decreases as a function of time when animals are engaged in a wide variety of tasks in a large range of neural areas. On the other hand, our task is quite unique in that the way we design the stimulus encourages the animals to integrate random samples of evidence over time. Here in this movie, I've showed you the VARCE computed from neural data as a function of time. This black trace shows the VARCE, and what I hope is apparent is that it increases dramatically over the time period when the monkey is forming his decision but has not yet committed to a particular choice. We then computed a second quantity from the data, the correlation of the conditional expectation, or the CORCE. We again began by looking at the spike counts in small counting windows, which I've shown by this black rectangle here. We then computed the spike counts in a neighboring window, which I've shown as a green rectangle, and looked at the correlation between the spike counts in those 2 windows. We did that for all subsequent time windows, and have shown the magnitude of the correlations below using this heat plot. Red squares indicate conditions where the correlation was very high, and blue squares indicate that the correlation was very weak. And you can see that the magnitude of the correlation decreases as a function of time. We then repeated this process for the next time window, generating another row of correlations, and so on throughout the trial until we have an entire CORCE matrix. So we're now armed with these 2 tools, the VARCE and the CORCE. We knew what they did in the data, and we decided to explore the predictions for these quantities from a whole range of theoretical models. And this turned out to be really interesting. There had been previously a number of candidate models for decision-making which we had been unable to distinguish because they all made identical predictions for the firing rate mean. But by using our new tools, the VARCE and the CORCE, we realized that these putative models for decision-making made very different predictions for the VARCE and the CORCE, and we could compare the model predictions with what we actually saw in the data. And this allowed us to rule out several models of decision-making which we had been previously unable to distinguish. There were, in the end, several models which successfully predicted the VARCE and CORCE that we saw in the data. And these models had in common that all of them involved integrating random samples of evidence over time. What I've shown in this figure here is evidence that is accumulated over time up to some threshold level or bound. When I overlay many such examples shown by the gray traces, what you can see is that the trial-to-trial variability, the variability across the traces, grows as a function of time, just like our VARCE did in the data. And similarly, you can imagine that the value at any particular time point is simply the value at a previous time point with one more value added to it. And that explains the long correlations that we saw in the CORCE. I'll finish up by saying that we hope these quantities, the VARCE and the CORCE, will be useful in a broad variety of contexts. We wondered whether other groups might find, as we did, that by looking at the trial-to-trial variability, that one can find much more information about neural mechanism than is apparent simply by looking at firing rate means.